The only aim of life of sperm is to unload, to safely transport and unload its genetic material to the ovum, right? And in this way it will form the zygote, and zygote will form a complete human being. Well, for this task, this is well suited. Sperm is well suited for this job. How come? First of all, it has to travel all the way. Sperms are loaded, lots of millions of sperms are loaded as an ejaculate after the sexual intercourse near the cervix, near the opening of cervix into vagina. And from this opening of cervix in the vagina, this sperm, I am showing only one sperm. The sperm has to travel all the way from this into this oocyte to the ovum that is wandering in the fallopian tube, right? So it has to move through this path, and for this it has a special tail, right? And with the help of this tail it will move and it will reach to the oocyte, right? Now here, as it reaches the oocyte, it has to face another problem. It cannot still transport its nucleus into the cytoplasm of oocyte. Why so? Because oocyte is guarded, it is protected by lots of follicular cells, right? It is surrounded by a lots of follicular cells that forms this crown-like structure, and this structure is called corona. What is this? Corona radiata, right? And not only it is surrounded by corona radiata, but it is also surrounded by this yellowish thing here. This what is this called? It is called zona pellucida. It is a glycoprotein coat. So this sperm has to penetrate through this corona radiata as well as zona pellucida in order to fuse with the membrane of oocyte, and after fusion it will then transport its nucleus into cytoplasm of oocyte to form zygote, and this we will discuss the details in fertilization lecture, right? Now how it can penetrate these layers, through these layers? Fortunately, it is provided with a special sac, a special sac at the tip of, a special sac is present at the tip of sperm or spermatozoa, and this sac contains a lot of proteolytic enzymes, right? And when these proteolytic enzymes are released here, these follicular cells, they separate from each other, and this zona pellucida, it breaks open, right? And in this way this acrosome helps to pave out the path for the sperm to unload, it's to fuse with the membrane of oocyte and to transport the nucleus, right? And of course, along with acrosome, acro mean tip, some mean body, it is present at the tip of sperm, so it is also called acrosome. And of course, along with acrosome, it is having flagellum or tail, and this tail is also called axoneme, and this tail helps the sperm to move all the way from cervix up to the fallopian tube or uterine tube. And for along with that, it is also provided at the base of tail, it is also provided with a lots of mitochondria that surrounds this tail, this axoneme, right, to provide energy for this voyage, for this traveling. It needs energy, and that is provided by mitochondria. So that is the structure of a mature sperm. What it contains: acrosome, nucleus, mitochondria and axoneme. And interestingly, this sperm is very small in size, and it has very few organelles that is very essential for it, and very little cytoplasm is present, and nucleus is tightly packed, condensed. All these things, it helps the it helps the sperm to easily move through female genital tract, right? So that is how due to these characteristic features, spermatozoa is very favorable for this job. And now we will see that how this spermatozoa forms. Right? In the previous lecture we had seen that spermatids are formed and they are still embedded into Sertoli cells. Right? So these spermatids, these are epithelial looking cell, epithelial like cells, right? And they do not look like sperm. They do not look like a spermatozoa in any way. They do not have tail, they do not have acrosome and all these things. So from these spermatids are differentiated, they are transformed into mature sperm or spermatozoa. And this process by which spermatids transform into mature spermatozoa, it is called spermiogenesis. So today we will talk about spermiogenesis. I have told you what is spermiogenesis, and now we will see how spermiogenesis occur. Right? Hello, I am Dr. Aizaz from MedicoVisual.com. Welcome to this visual lecture. So here we have spermatid and it is having a highly condensed nucleus, right? So first we will look that how acrosome forms, right? As I have previously told you that acrosome is a specialized sac that consists of lots of proteolytic enzymes. And you know from the previous knowledge that enzymes by nature are proteins. And how does any protein of the body forms? Proteins, the recipe to form protein, it is present in the nucleus, in the DNA. In the DNA there is specialized nucleotide sequence that tells the body that how to synthesize that protein, and that specialized sequence is called gene. That is gene of that particular protein. In this way, different genes of different proteolytic proteins or proteolytic enzymes of acrosomes are present in the DNA. And let's suppose this is a gene of any particular protein or particular enzyme of acrosome. From this, it is transcribed into mRNA, messenger RNA, right? And from this messenger RNA will come out of nucleus and it will go to, what is this? Rough endoplasmic reticulum. This is endoplasmic reticulum with ribosomes attached on its surface, and that is why it is rough endoplasmic reticulum. So mRNA will go towards the rough endoplasmic reticulum, and here these ribosomes will translate this mRNA into a protein, into polypeptide. So this newly being synthesized polypeptide will be entrenched into lumen of endoplasmic reticulum. It will enter into lumen of endoplasmic reticulum and will be further processed in the lumen of endoplasmic reticulum to form complete proteins, right? So in this way, acrosomal enzymes or proteins are formed in the endoplasmic reticulum, and then some part of this part of endoplasmic reticulum will bud out, right, and form a special sac, and we have Golgi apparatus inside the cell, and it will fuse with the part of Golgi apparatus, and Golgi apparatus will then pack these enzymes into special secretory vesicle. So here this is a secretory vesicle that contains an aggregate of lots of acrosomal enzymes, right? In this way, lots of small vesicles or small secretory vesicles or sacs of different enzymes of acrosome, they are formed by endoplasmic reticulum and Golgi apparatus, and they ultimately coalesce together. They fuse together to form a relatively larger sac that is called acrosome, right? Meanwhile, nucleus condenses, it further condenses so that it becomes easier to transport, right? And acrosome is arranged in front of this nucleus. Look at the beauty of nature. This acrosome is, this nucleus, it is strategically placed just behind the acrosome. So as it reaches the oocyte and it paves the way to unload the nucleus, this acrosome will be immediately used up, and then nucleus that is immediately lying behind that acrosome, it will immediately enter into oocyte. It will immediately be unloaded into cytoplasm of oocyte, right? So that is why it is strategically placed, nucleus is strategically placed just behind the acrosome, right? So this was how acrosome forms. Now we have a special organelle, that is centriole, and the centriole is present somewhere in the cell, and it comes and moves to that position of the cell from which tail has to arise or flagellum has to arise, and flagellum will arise from this centriole, right? Are you clear up till now? And we have lots of mitochondria as well. These mitochondria, you know, they have ability to self-replicate. They can regenerate mitochondria themselves, right? So lots of mitochondria, they will gather at the base of this tail, at the base of this flagellum, and meanwhile flagellum is being formed by the extension of this centriole. And these mitochondria, they are arranged at the base of flagellum, at the base of tail, right? Meanwhile, what is this? You can see that there is lots of excess cytoplasm in the sperm that it does not need, right? Mature sperm contain very little cytoplasm to make it lighter and easier to move through this female genital tract. And finally, this excess cytoplasm along with some organelles, it separate out. What happens to Golgi apparatus and endoplasmic reticulum? It is either completely used up to form acrosome and other proteins, or somewhat that is left behind. It is also, it comes here in this excess cytoplasm, and it separates out and it is eaten by Sertoli cells. You know, it is still the spermatozoa, it is still embedded into Sertoli cells. It has not yet came out. So Sertoli cells, they nourish this, they provide protection to these developing spermatozoa, but another function of Sertoli cell is to engulf that excess cytoplasm and excess organelles that it does not need, right? So this was about the spermiogenesis, that production of sperm, mature spermatozoa. Now this much information is sufficient for most of you guys, but I want to make it really clear that how this tail forms, right? So in next lecture we will discuss how this tail or flagellum develops from this centriole, right? First we will discuss what is the structure of centriole, then we will discuss how this tail or flagellum forms, and then finally we will discuss how it matures, and we will discuss all these things in a 3D software, right? Thank you so much for watching this video, and wait for the next part. Please give your precious comments regarding this video, and if you haven't subscribed, please subscribe to my channel.